All right, so 3 topics we're going to go over today. It's going to be the scientific method, then bioinformatics, oh my gosh, and then human genetics and diseases. Well, not really into detail, but it's more like a project, okay? All right. So let's start out with the scientific method. All right. I like this lecture. So, to start off, what exactly is science?
Oh, no, you did not.
All right. So if you were to search up science on Google, okay, you'll find out, it'll spit a couple things. It'll say, it's a study of the natural world through observation and experiment. What does that mean? I mean, observation and experiment, that makes sense, right? But the thing about that definition is it's somewhat incorrect. Yeah, I know, the world got it incorrectly. That's crazy. Because it's missing something very valuable. For me, in this class, I want you guys to define science as 2 things. For sure, it has to be testable. That's for sure. But it also has to be falsifiable. And we'll go over why that is, okay? In a couple more slides. But let's talk about why we study science in the first place. And why is it so important? Especially to begin off understanding it before we start a lab. Okay, we're all doing scientific research here. Okay? So why do we study science in the first place? So I want you guys to go ahead and stare at this frame. Raise your hand if you see a lion. All right, nice. That's awesome. Now, imagine this was, let's just say this was maybe 60,000 years ago, during our caveman years, okay? I don't know why I'm doing this, but during our caveman years, there you go, like this, right? There we go. And you stumble upon something like this. Oh my gosh. Raise your hand if you run away. Wise move. Now raise your hand if you're like, it could be a lion, but then again, it could be my friend playing a trick on me. No? Okay, wise, very wise. Now raise your hand and you're like, it's just a lion, I could beat it up. No? None of you guys? Maybe not. All right. So those of you guys who decided back then, 60,000 years ago, and decided to book off like a chicken, guess where you guys are today? You guys are alive, and you guys are now the descendants, the ancestor of everyone here in this room. Crazy, right? You guys were able to survive and pass down your gene, the chicken gene, and to the point where you're even today you even are able to run away. Now, what makes it even more unique is if you take a look at this lion, what's the first thing you guys notice about it? What do you guys notice about this lion?
Hungry.
The tongue, the face. What else do you guys notice? The first thing you guys notice when you look at this picture. It looks like it's camouflage in there. You look at the eyes. So most of you guys recognize the facial area, right? That's like the first thing you spot out. That's very interesting. So the instinct in which you're able to pick up something that's hiding, and the first thing you know is the facial expression, is so keen for your survival instinct, especially back then, to the point where now it's embedded into our own genome. Who here see faces on these objects? You guys see faces? Those aren't faces though. But you guys see it. That's crazy, right? The reason why you guys are able to see it is because your survival instinct, it's just so strong that it's embedded into your genome that you guys now recognize those faces, even though there's no faces there. But I mean, think about it. Who here has friends or cousins or siblings who pop up and scare you? You know, like you hate that, right? But instead of saying, and then cussing at that, like, screw you, man. Just like, thank you for testing my evolutionary instinct. I still got it. Right? That's what it is. So again, the reason why we study science is because the consequence of our evolutionary bias is kicked in. And so we see things and we believe in things that maybe are not there. Okay? And so because of that emotion, that instinct that we have, it can mislead us to fall for horoscopes. I mean, who here believe in horoscopes? None of you guys? Okay, well, some of you guys. My wife deeply believes in horoscope, and she's gonna be a doctor next year. So, but there's still people who believe in it. That's fine. There's fortune tellers. There's scam artists, salespeople. I mean, every time you see something like, it's so funny. Bed Bath Beyond. You know how they're like selling everything off? It's like 50% off by default and all that. Me and my wife went there to buy something. We look in there. We're like, oh my God. They're like, oh yeah, anything you purchase here, the original price, once you go to sale, we'll remove 75% of it. We're like, oh my God, that's a steal. We look at the price, like, did they jacked up the price 80% first or something? Because we saw like overpriced first. I was like, wow, those are premium price. So that's how they trick you. But a lot of people fall for it. They're like, oh my God, big sale, and they fall for it. Same thing for people who sell cars. Like, oh, this car is in really high demand. We have to charge $5,000 extra. Otherwise, another person will get it. See that guy right there? He's going to get it, right? And they trick you to buy it, right? And same thing for a lot of things that we do. How a magician and illusionist, right? Who here listen to music? I'm pretty sure all of us do. But we don't listen to random music, right? Like for example, let's just say you're going clubbing and you're listening to like classical music. It doesn't work, right? So
Perfect.
I know. So you listen to music that flows with your emotions, right? So certain songs that talk about love songs or happy song, sad song, you follow that emotion through and you feel the emotion. You feel like that song is talking about you, right? And that's normal. That's fine. And then who here believe in ghosts and monsters when they're young? Come on, none of us? If you don't believe in ghosts and monsters when you're young, you have a mutation somewhere. And the reason why, imagine you're a kid, 60,000 years ago, did not believe in ghosts or monsters. And as a kid, you are a curious person. What do you think would happen to that kid when he roamed by himself, sneaking out into the wilderness?
I think it'll be fine.
It would be fine. Or something would eat it, pick it up, right. That kid would be a goner if it doesn't believe in ghosts and monsters. And to the point where some of us still, believe in ghosts and monsters even today. Even at our adult age. It's still embedded to us. Right? Alright. So that's just gonna give you a better idea of why we have to apply science. It's mainly because we're biased. But I haven't really answered the question why it has to be falsifiable. I mean, I just said we have to do science, right? But what does that falsifiable mean? Why science has to be falsifiable? Can you do science on everything? And the answer is not quite. And I'm going to tell you a story. It's called the Red Dragon Hypothesis. You guys know who Carl Sagan is? Anyone? Okay, Carl Sagan. Some of you guys know him. Most of you guys maybe a new name. Carl Sagan. You know how about Bill Nye the Science Guy? You guys know that? All right. Most of you guys do. Okay. So Bill Nye the Science Guy, he's a very famous, TV, television scientist in the 90s. Very famous. Carl Sagan is very famous in the 80s, just a decade later. Okay. And Carl Sagan came up with something very unique. It's called the Red Dragon Hypothesis. And the Red Dragon Hypothesis goes something like this. All right, we're gonna do a play, all right? So I have a red dragon. Okay, you guys want to check it out? All right, cool. You guys want to check it out? All right, I bring you over. It's like, all right, cool. This is my house. I open up the garage, and you don't see anything. And I just tell you like, oh, I forgot one more thing. He's super shy, so he becomes invisible when he sees strangers besides myself. Okay, so at this point, you're like, wow, this guy's crazy. But you're curious. You're going to ask me any question you want. Okay? It doesn't matter what question you want. Go ahead and ask me any question. What question will you ask?
What color is he?
What color is he? Oh, he's a red dragon. That's like I said, red dragon.
Can you take a photo of him and show him to us?
He's very camera shy. He could sense the electronic from the camera, so he'll disappear right away.
What gender is he?
Oh, it's a he. It's a he.
How do you know? Have you checked?
No. Any other question you may ask?
What happens if we can see him through a mirror?
Can you see him through a mirror? Again, he knows there's reflection and you'll hide away from it too. He just senses it. He has that hormonal sense that I was talking about. That's a good question. Well, and it just so happened that he likes me. So that's why he only showed up for me.
Where'd you find it?
You know, one day I was, very sad in the rain and it appears and I'm happy again. So the point of this particular hypothesis here, the red dragon hypothesis story, is to tell you, you could ask me, honestly, you could ask me billions of questions all day, forever, for us, eternally, but I will always come up with billions of excuses. So in other words, you cannot do science on something that is not falsifiable. That makes sense? Which is why it's so saddened for me that on Google, when you look up what is science, it doesn't show the word falsifiable, which is kind of saddened. Because in reality, you cannot do science on something that's like that. That's not falsifiable. Infallible, right? Cannot do that. And that's how people get mixed up between science and beliefs, science and faith. It doesn't work that way, right? Yes.
So that's why there's like no research on like ghosts.
Yeah. It's infallible. We cannot do it. And whoever's doing that, they're just weird. They're not really true scientists. They're just doing that for the fame of it.
There probably is people doing that, right?
Yeah, there's probably people trying to do that, but it's not really scientific.
But at some point, didn't we think some things weren't scientific, and they ended up proving it, and we were like,
If you ask the right question, and you're able to answer that particular question, that's perfectly fine. But when it comes to something that's infallible, you cannot really do science on it. Because you could come up with billions of other excuses justifying why it didn't work, or why it did work, right? All right. So how exactly do we protect ourselves from these kind of biases? Well, we will protect ourselves through something called the scientific method. This is a great way to protect yourself. And the order of the scientific method is through something like this. Observation is something we observe, like, oh, I see that, I don't see a trash can, but can someone bring the trash can right in the middle here for me? Thank you. Thank you. Let's bring it right in the middle. Let's all observe that trash can. Here's my observation. I see that trash can. All right, that's my observation, all right? Let's all see that, all right? You guys all see it? All right, so it's not infallible. Cool. My question is, how many feet do you think that trash can is? How tall is it? That's my question. And next, it's going to be a hypothesis. So I want you guys go ahead and discuss among each other really quick. Go ahead and shout and answer. How many feet is that?
who knows their height here? One Joe small.
Hold on.
What do you guys think? Come on, guys. Have a hypothesis.
Okay, I think it's like 3.
Like 2.5, 3.
3 feet, all right. 2.5?
I think it's like
I think it's 3.
2.5.
2.5?
I think
All right.
it's
3? All right. Excellent. So,
I want to know now.
I want to clarify this to you guys. Everything that you've been taught in regards to come up with the hypothesis, I'm not sure you guys know this, but were you guys ever taught if and then?
Yes.
And you guys still apply that when you guys make a hypothesis? Somewhat?
Yeah.
For some reason, it just complicates everything, to be honest. Every time a person uses if and then, it complicates everything. However, I understand why. The reason why they use if and then is because they don't want you guys to assume a hypothesis is a conclusion. That's the reason why. But I want to make sure that you guys don't mix up hypothesis and conclusion. So in other words, I just deleted conclusion altogether. Okay? So you don't need to worry about conclusion, because in regards to hypothesis is literally your most educated guess. And you guys did an educated guess. You base it on something you know, like Jacob's height, and then you assume it's 2.5. That's your hypothesis. Like, it's an answer. A hypothesis is basically an answer to a question. It could be invalid, it could be something bizarre, but it's still a hypothesis. It's your most educated guess. Some of you guys just eyeball it 3 feet, you know? And that's your hypothesis. So a hypothesis is basically an answer to a question. That make sense? All right, and then afterwards, you can then do an experiment. What kind of experiment can you do? Cut chicken in half. No. No one's gonna use a ruler? No? A measuring tape? A measuring tape, right? So then you can start testing it and validate it. Right? And then, of course, here's the thing. A lot of us still fall for a trick, right? Whenever, and this is just very straightforward. Like I'm literally telling you, hey, come up with a hypothesis. Here it is, right? I'm literally telling you. But in reality, in TVs and in real world, you're not going to have an instructor like Dr. Pham telling you, hey, come up with a hypothesis. You guys will have to formulate that yourself. And sometimes, most of the time, you guys never do. You jump straight to conclusion. What do I mean? Let's take a look at this example here. Observation. You're looking at a YouTube or something, and you saw a magician doing an awesome card trick. Go ahead and take like 5 seconds there, and just think about what's going on there. That's pretty cool, huh? Like, how's doing that? That's awesome. That's crazy. That guy up there, he's a very good magician. All right. Now that we observe it, what's in your mind? Tell me something that's in your mind about this.
Sleight of hand.
What's that?
Sleight of hand.
Sleight of hand. What else?
It's magic.
Is magic. What else? What's that? You're still trying to figure it out. Okay. All right. So most of you guys just went straight to conclusion. That's crazy. And none of you guys asked questions like, how is he doing that? Not in your mind, right? So a lot of people are like that. A lot of people, when they watch something or see something, they go straight to conclusion or even giving their own judgment about it. But they never ask a question. So that's the thing I want to train you guys on. At least get used to it. Okay? Is to apply the scientific method. Remember, it follows this step. Observation, what you see. Question, hypothesis. And then your experiment. Okay? So, you guys ready for it? You guys want to test it? All right, let's test it. All right. Imagine this.
Oh, you did.
You like that, right? All right. Imagine this. Imagine you're on, you're watching your YouTube channel or wherever you're trying to watch, and then a commercial popped up. It's really weird, and you're like, oh, cool. Like, oh, it's just in. We have discovered that dihydrogen monoxide is found in all ocean toxin and pollutant. We also have discovered, based on our research, is that high dose of dihydrogen monoxide is extremely toxic to the human body. Please vote yes to ban and purify dihydrogen monoxide in all our drinks. Just hearing this. Showing up like science, biohazard, everything, and it just sounds so serious, right? So, in your mind, I want everyone to raise their hand. Thumbs up and thumbs down, okay? I want everyone to participate. Who here would agree to purify the drinks from dihydrogen monoxide? Thumbs up, thumbs down. Go ahead. Thumbs up, thumbs down, thumbs down. Okay, I got a few students putting thumbs up. All right. So, that's kind of weird. Why you guys want to purify our drinks? Oh, sorry. Thumbs up is purify. That's good. So the rest of you guys don't want to purify dihydrogen monoxide? I mean, carbon monoxide could kill us, but you guys sure you want to get rid of dihydrogen monoxide? Why not? Tell me. It's water. What? What do you mean it's water? Tell me. Dihydrogen monoxide means water, but you guys voted yes on it. Why?
Because I assume the information I got was factual.
You assume the information is factual. Oh my goodness. It is toxic. Oh my goodness. Wow, you fell for the trick. As you can see, we as a human, I mean this is just a satire. Or is it? No, it's a satire.
Oh my god.
And this satire right here, I just want to show you that there's literally commercials that go beyond things like this. Like a lot beyond like this. This is just a satire, but simplistic. But there's a lot of commercials that you may watch, especially in politics, that may show a film that sounds so factual, that tricks your emotion to believe in it, and you vote against your own, you vote against yourself. That makes sense? And a lot of times that's what happens. A lot of people And here's the thing, people who are in commercial field, advertisement field, those are smart people. Like, I would say they're equivalently smart as me, you know, like, or any other doctors. But they're really skillful of manipulating a human in their mind and thinking. It's just like yourself. You were tricked to believe that's a fact. That's crazy. And so a lot of commercials like that. And the thing, how exactly do you defend yourself? Well, for one, you have knowledge. Like, some of you guys already know what nitrogen monoxide is, but what if you don't know? The first thing you should do as a scientific method is, instead of jumping straight to your emotion, you search up for it. What exactly is nitrogen monoxide? How is it poisonous to us? What do you mean by high dosage? Is it really found in it? In ocean? You know, then you search for all answers. You search it for yourself rather than come up with a conclusion. Right? But it's okay. Remember this, we're all human. We are born to be biased. So it's okay to be emotionally biased, to believe in things, because that's what makes us human. If you're bland and all that, you're unique. Okay? But otherwise, we're born to be unique. We're born to be emotional with our own biases. So it's okay that you fall for a trick. It's okay to jump straight to conclusion or judge somebody without asking questions, because that's what makes us who we are. But in this class, to be thinking like a scientist, I want you guys to be very careful, because sometimes our emotion may lead us down a rabbit hole that may not be good, right? So the first step in the scientific method is to ask the question. Okay? All right. Always ask questions. So that being said, go ahead and take the next 2 minutes. Look at these questions here, because you can't just ask any question, right? So go ahead and come up and discuss among each other which one is a good question and which one is not a scientific question. So go ahead and discuss. Take 2 minutes. All right. Let's go over this together. So, table 1. Will having trees in my backyard cool down my house? Is that a good question? Yes, that's a very good question. It's testable and it's falsifiable. What about this? Table, you guys, what do you guys think? Is my friend possessed by demon? Is that a valid scientific question?
No.
No. Excellent. Although it is testable, it's infallible. Very good. All right, back there, what do you guys think? Do houseflies tend to swarm around dirtier areas? Yes, that's a valid question. And finally, but not least, back there, should I pray daily to improve my test score? Is that a good, valid question? No. Very good. It's testable, although it is testable, that's for sure, but it's not falsifiable. That makes sense? All right. So, even though the first step of scientific method is to ask question, you have to ask the correct question that is valid to be scientific. Make sense? All right. Cool. So after, let's just say you came up with a great question, then you come up with a hypothesis. Remember earlier we said here, you're basically giving your most educated guess on how tall that trash bin is, or trash can is, right? And most of you said 2.5 over there. Some guys just eyeball it, 3 feet, right? That's your valid. That's a very valid hypothesis. A hypothesis is basically your most educated guess. And all you guys did was hit 2 feet, 3 feet, 2.5. It's just literally a number, and that's a hypothesis. And that is a valid hypothesis. It's not a conclusion, remember? You can say it's 3 feet and conclude that, but I don't want you guys to conclude that. It's still a hypothesis. All right, that's why we remove a conclusion there. We want to be as scientific as possible. Now, how exactly does a hypothesis eventually become a theory or a law? Well, a theory is a unifying explanation of a set of verified proven facts. Okay, it's kind of weirdly put, but let me give you an example here. Imagine this. How do we turn 2.5 feet into a theory? It's when we measure it over and over, every single one of us measure it, then we give to every single individual around the world, the expert, to measure it, and they're like, yeah, it's still 2.5 feet. Yeah, and then some person in Asia, yep, still 2.5 feet. And then some person in Africa, yep, it looks 2.5 feet to me. And there's no other reasons that not 2.5 feet. Like, it's just basically have all the evidence for 2.5 feet. And every single experiment they do is still go back to 2.5 feet. So because it's just a strong evidence, no evidence so far has disproven it, we can then claim that to be a theory. It's so strongly backed up. And that usually takes time to become a theory. So, another word, the word theory in science is very powerful. There's a very powerful meaning to it. So when we say the theory of gravity, right now it's called the law of gravity, but when we say the theory of gravity, it's just so powerful that no matter what, you know it's a known fact. With theory of evolution, no matter what, it's a known fact. You cannot deny it, right? It's 2.5 feet. No matter what, that's 2.5 feet. You can't really deny it. You can make up some other things to try disproving it, but it's still going to end up 2.5 feet. A theory is that strong. It still end up to be consistent all the time. All right. All right. So a theory has a very strong predictive power. And that being said, we're going to use that theory of evolution in our next activity. Okay? Well, we're going to apply. But before we start and deep dive into bioinformatics, let's go back to the scientific method. Because I'm assuming now that you listen to my lecture, you guys are now experts, right? Right? Yeah, you guys are. Okay, good. I want to be confident. All right. So that being said, let's start out with observation. This is what you observe. You're mixing this together. And the question for you guys is, what color are their offsprings? And this is going to be all your observation, okay? But let's start out first. What would you be your best educated guess if you were to mix a red and a red?
Red.
Red, right. So it'll be a red. Excellent. That's an observation you saw. All right, that's pretty easy. Well, what about if you mix white and a white?
White.
White. Let's see if you got that correctly. Oh, it is correct. Very good. All right. Bonus question. Let's see. What if you mix a red and a white? What would be your hypothesis there?
Pink.
A pink? Is that right? Anyone else got pink? You guys got that? All right. Let's see what you guys got. And oh wow, it's also a pink. Look at that. You guys got lucky. Otherwise, it could be a different color. Good job. All right. What about this? What if you got if you were to mix a pink and a pink?
Pink.
Based on all your observation there, you got pink. Anyone else? You guys all agree with pink as well? All right. Let's see the answer. Assuming that you threw in 50 cents in there or something, that you place a bet. You don't get pink. You, for some reason, get all 3 colors again. Holy cow. So your hypothesis was wrong, but don't worry. Back in the 1800s, they also cannot figure this out. It took them many, many years, hundreds of years, to actually figure it out. And the person who helped figure it out is Mendel, Gregor Mendel. But don't worry, just like you guys that came up with and hypothesized pink, you get a law scientist got stumped too. I mean, look at that. He's like, whoa, how? And this cat is like, whoa, no way. Right? Even they were surprised. Like, how is that possible? So how is it possible that somehow you mix pink and pink and it becomes all 3 colors again? Or sometimes when you mix pink and pink and it becomes red. What? Or when you mix pink and pink, your offspring becomes white. And just white. How is that possible? What's the mechanism behind that? So there's still, now I did show 3 images here, but what makes scientists back then so confused is that sometimes they don't see all 3, they only see red only, after you mix pink and pink, and that confused them. Or sometimes they only see white, they don't see this too, you mix pink and pink and all they see is white. You're like, wait, what? That's confusing, right? So the more you do it, sometimes you start seeing they all produce all 3 colors. So how exactly is that even possible? Well, from Mendelian inheritance, what it is that we share 50% of our alleles, our traits, from our mom, and 50% from our dad. You guys agree? Right. So 50% from mom, let's just show in red, and 50% in dad, let's just show in blue. And therefore you is 50-50. You see that? All right. We can assign your alleles with color code letter. Okay, so I just basically assign the alleles with color so you guys can envision what's going on. All right. And then another thing, what Mendel had discovered and he defined is something called the law of dominance. What exactly is the law of dominance? The law of dominance basically define if there's going to be a certain trait, we can define this trait as a capital letter. If the trait is a capitalized letter, okay, then that one trait is going to be expressed no matter what. Even if you have a lowercase letter trait, you still can be showing all red, just for example. And however, if you have only way to show the other color, let's just say the white color, which is showing lowercase level, the only way to get a white color is if both your alleles are recessive or lowercase. So in other words, even if there's one dominant trait, no matter what it is, even though you have a bunch of lower letters here, that one trait is the only one that's going to show. That's the law of dominant. And Mendel got very lucky that he was studying something called complete dominant traits. So the only way this was possible, remember, they were trying to figure out and understand how genes and traits are passed down for hundreds of years. And Mendel was very lucky because the crops, the traits that he was studying are dominant traits, right? A lot of scientists, they were studying incomplete dominant traits, which makes them very confused. Okay? So let's talk about it. So let's do this. So complete dominant is basically when they have a capital letter, it will dominate the whole entire expression. So if you have a capital R, then it's going to be all red. If you have a heterozygous, regardless, even if it's a lower letter, that capital letter overwhelms it and still show red. Until you have only lowercase only, then it'll be white. Okay? That'll be the law of dominant. That makes sense? So you can see this picture. Red plus white doesn't matter. It'll be red. Okay? In regards to incomplete dominant, it's slightly different. It's what we follow the blending theory. So it has to be capital letter R will be red. If you have a capital R and lowercase r, which is heterozygous, then it will be a blend of the two. It will be a pink. And then you have lowercase r and lowercase r, then it'll be white, right? And that's the example I gave you earlier. And finally, but not least, there's something called codominant. Codominant is basically when both gene and both traits are very strongly expressed. So capital R, in this case, it can be W. I just put W there for white. So RR for all red. RW is for both traits. You show both red and white. And then there's W and W, which shows all white. And a good example of a codominant will be your blood type. Blood type A, and then blood type B, and there's also blood type AB, right? And that would be a good example of your codominance. Do I have you guys do Punnett square? I don't. You guys know how to do Punnett square? Okay, I'm not required to teach you guys Punnett square. So, okay, I'll just show you one example, okay? I'll show you guys, let's see, this guy right here, the last guy, pink. You were to mix pink and pink. How exactly does this come up with all three? Yeah, right? Pink and pink. Oh, I don't have it. But the question is, how exactly does this become all three? So I'll just show it to you guys. Biochem. There we go. And then I'm going to use... Oh, wow, convenient. I like this. All right. So let's just say you have your mom. And your mom is heterozygous, R and R. And she's pink, right? Oh, cannot see it. No. Let's do this. Markers. Much bigger. Mom is R, big R and little r. It's much better. And dad is the same. Big R, little r. You see that? So the way you apply the Punnett square will help you define what's the chance that your offspring will be red, what's the chance it will be white, and what's the chance it will be pink. That's basically what the purpose of the Punnett square is. They give you statistics of your offspring. Okay? So the way you do it, you're going to draw something called a Punnett square, which is basically a box with 4 boxes in the middle, inside of it. Just like that. You guys seen this before. And what I'm going to do is that I'm going to drag mom to the side. So here, mom right here. And dad's going to be on top right here. R. This big R come from this. right? This little r come from the little r over there, right? And then same thing here. This big R come from this big R of dad, and this little r come from this guy. Got that? So now we could drag it down. I'm gonna drag down dad first. So R here. I'm dragging down all the big R's, and then I'm gonna drag down the little R's next. Little R, little R. See what I did? And now I'm gonna put mom across. Big R, and then little R here, little R here. So now you get to look at it. All right, there's one big R, right? It's like what, 25% chance there's gonna be a red offspring, and there are 2 heterozygous, so 50% chance will be pink, and there are 1 little r, a homozygous recessive. In this case, it's 25% chance to be white. And that's how you come up with statistics to determine what's the chance that you can have this kid with this trait. Okay? And that's how you do the Punnett square. And that's the mechanism how nowadays, in the modern days, we understand how traits are passed down from parents to offspring. Okay? And you can actually do this for a living if you want to. There's people who do this for you for a living. All right. Any question about that? Was that clear for you guys? All right. You'd be surprised when I teach the high school. It takes me 2 weeks to have them understand that. You guys know it's true, right? But it's crazy. But you guys are good. They're the good students. Man, I get really frustrated when I teach high school. If you plan to teach high school, you gotta have a very strong patient. All right. All right. So genetics. So that was Mendelian genetics. So Mendelian inheritance, they taught us a couple things. We already just went over the law of dominance when we learned about dominance and recessive traits, right? So we just briefly went over that. Another thing as well I want you guys to understand is phenotype versus genotype. What is the difference between the two? And also, what's autosomal and sex link? So go ahead and take the next 1 minute, discuss among your table. Go ahead and do that. And describe what's phenotype, what's genotype, what's autosomal, and what's sex link. What exactly are those? So go ahead and discuss. Let's go over this together. So, phenotype. What exactly is a phenotype and a genotype? What's the difference? Can anyone tell me? Okay, the phenotype is the phenomenon, the appearance of what's shown. It's the phenomenon, what you observe it. Very good. And what's genotype?
Genes?
It's the genes. It's the traits, the alleles. Basically, it's the genome. A, T, C, G. Right? That's basically what the genotype is. It's a sequence. We could simplify it saying capital A, lowercase a, right? Those letters. We could call those as genotype too if you want to. Okay? But it's basically the genome. That's the genotype. And phenotype is what you observe. Very good. What's the difference between autosomal and sex link? Yes?
Sex link is that links to the actual sex gene.
All right. Very good. So the sex link gene or chromosome will be this guy only, the X and Y. Thank you. It will define whether or not you are a male or a female or both. Right? And then autosomal traits would be all the rest, the body chromosomes, right? The rest. Okay? And when you see something like this displayed for you guys, they usually organize it based on size, from big to small. That's how they organize it. So if you're curious, like, how do you know which chromosome is where? They base it on their size. And that's how they map it. Actually, there's a company that does this for a living. They make trillions of dollars just this is what we call karyotyping. And whenever you're, a lot of time, especially when you're pregnant, especially when you're like a higher age, it's very important. I highly recommend it. Get your placenta, your baby, karyotyped, because you want to observe whether or not there is some abnormality. Because the most common one will be a chromosome 21, could be triplicated, right? Have 3 of them, which lead to Down syndrome. Or another one is triplication of chromosome 19. I think that's like Turner syndrome or something like that. So you gotta So when you're pregnant, and especially at an older age, like 35 or something, that's what they call it. Always double check because you have a higher risk for this to occur. Okay. All right.
Can they do it in adults too, or just on babies?
Adults?
Yeah, can adults?
Oh yeah, you can always keratote yourself. That's a lot of people do that. Even I keratote myself when just for fun. You get to see your chromosome. Yeah, it's pretty awesome. If you guys want to do that in the biotech lab, we could do that. Keratote yourself. All right. Let's get to this. Let's do this. This is the last one, guys. Bioinformatics. You guys heard of that before? How many of you guys did bioinformatics? Raise your hand. Damn, that's a lot of you guys. Nice. Good job. So what exactly is bioinformatics? And have you ever done bioinformatics? So I know some of you guys maybe do a bioinformatics going online, looking at genomes and all that. But in your regular daily basis, all of you guys do bioinformatics almost every single day without even realizing it. You guys know that? Who here search up definition of medical terms? That's bioinformatics. Whoa, you did it. Who here look at YouTube videos to learn? I'm pretty sure most of you guys do. That's bioinformatics. Bioinformatics is basically computer-based research. Whenever you're learning or finding something new on a computer, especially about medicine or something related to biology, that's bioinformatics. That makes sense? So you guys are already doing that in your daily basis. Crazy, right? But today we're going to go into slightly something different. We're going to go towards proteins and study more about proteins, okay? But why bioinformatics is so important? Well, first, it's a computer base, and so it's very quick. You get your answer right away. Instead of going to your library, searching all up, or going to your professor and annoying that person the whole time and asking for information, you can just go on computer and get the answer right away, right? Just like that. And on top of that, it's cost effective. It doesn't cost you anything, just besides internet money, like a few cents for electricity, but it costs like nothing, right? Just to search up. So that's a huge advantage of bioinformatics. So today, what I want you guys to do, this is going to be a practice for bioinformatics, and then we're going to go on to your project, okay? You guys go ahead and get your laboratory manual up. I think it's on page 6. I put here. I could change it, but I'm assuming it's on page 6. Go ahead and go to page 6 in regards to your laboratory manual. And we are going to do the following. You see here, it's an example, earlobes, attached earlobes, right? Anything that's underlined, it italicized, it's going to be recessive, like little r, for example. And anything that's bolded, that's dominant trait, okay? So I give the answer for you guys there, okay? And then description of it, earlobe hanging, earlobe attached. And then it all has, in your table, it should have all the OMIM number, right? In this case right here. And then what you're gonna do for this particular exercise, I want you guys to go ahead and gather around your, I'm not sure what I say, observe your phenotype, count the number of class phenotype of the classmates. So first, you're gonna observe your own phenotype. Do I have this or do I not have that? Okay? If you have no idea what earlobes is or what free hang is, this is when you do bioinformatics. Go on Google and search it up. That's bioinformatics, right? If you have no idea what earlobes are, it's this guy. Okay? And then you're gonna check your phenotype. Are you recessive? Is it attached? Like for mine, I'm a recessive. Or are you dominant? That you see a hanging, right? You put a check there. After that, you can put down this. I mean, this should be easy. Big E, little E, to all of them, just like that. And then at the end, after you check your phenotype out, you can check every single one of your classmates and put down the numbers. How many class display, free hanging, and how many people display attached. And you can do that for all the traits in the laboratory manual. You guys got that? And if you're wondering, the purpose of inputting this number here is when you input it into this website, it's not going to shoot out a genotype or anything. It's just going to tell you more information about it. So you're looking up the history about it or the study about it. It's just going to give you more detail on how they describe it. Okay? That's what the OMIM number do. Right? It's just give you more information of what it is. All right. So don't get tripped out. It's like, oh my God, this bunch of number. It's just a reference number saying like, oh, if you're wondering what earlobes look like and how to describe it, use this number and go on this website. It will tell you everything about it. So it'll give you more description. Cool? All right, so go for it. Take the next 20 minutes and finish up that table. And this is where it all begins. So everything from this point on, make sure you save all those data down because you'll use those data again to formulate your project, okay? You guys have the slide. You don't need to take a picture of it, but I guess that's fine. Okay. I said here, find a partner. I think the rubric, let me check the rubric. What did the rubric say? 3 or 4? Okay. So don't worry about the partner thing. You're just going to do it within your current group. You guys are satisfied with the group you guys have? You guys good? Okay. So, within your group, what you're going to be doing is that you're going to choose one chromosome of the 23 pairs. Each of these are different chromosomes. You see that? So you're going to choose one of them. It can be any one of them, or it's up to you how you want to do it. But you can choose, basically you have to choose one chromosome. From that one chromosome, you're going to find one disease under your chromosome. And then this is just a summary. I think I have a picture to summarize it. And then from there, after you choose one disease from that one chromosome, determine that one causation gene. What gene may be linked, or a mutation that may be linked to cause that disease. Okay? And then you're going to read through page 7, follow all the order, what you need to do, and fill out that table on 1.2. Okay? Basically, that's the project that you guys are doing. So let's see. Oh yeah, I do step by step. So step 1, you're going to choose a chromosome. For example, let's just say you just love the number 6 for some reason, or whatever number you love. You're going to type down on Google, chromosome 6 disorder list. Okay? But make sure it's, be aware, some gene you may choose, like Down syndrome, that's not really because of a gene. That's a replication of a whole entire chromosome. So like, don't choose those kind of, don't look at replication like that, okay? So look at like mutation, like what mutation is causing a disease. More like that, okay? So you can choose a chromosome, disorder list. It's gonna list out all the disorder. You could basically randomly choose one of this if you want to, or choose the one you like. I look at this, hemochromatosis. It just sounds cool, so I just chose that as a disease. So choose one disease. And then afterwards, you can complete table 1.2. All of this guy here. Your group member, I was by myself, so you see that there. But you guys have a whole group. You can choose, you write down your chromosome number, your gene that causes it, right? So this HFE is the one that causes that disease, okay? The disease name or phenotype, hemochromatosis. The mode of inheritance, well, I chose chromosome number 6, so it's going to be through autosomal. And I did research on it. It said it to be a dominant gene, right? It's not a recessive gene. And I look at the base pair. How long is it? It took me a while to look at this, but it took me a while, so I just went on Google and it shoots up that number. So I just chose that number. All right, so it's pretty big. And then the typical life expectancy of that person. So it's like, okay, age. The survival rate is like 76%. That's basically if you have that disease. And then what's the common symptom if you do have the mutation by accident? What is it? Fatigue, joint pain, you're sexless, and those little things, right? So it's kind of give you a better idea. This is what the table I want you filled out. So that being said, go ahead. Oh, let's break again. Go ahead and fill that. Go ahead and work together and find that disease and fill out that table. Let's do that together as a group. Make sure you guys make sure this tab right here said protein, okay? Don't use another tab. And then input your gene name. So if you have your gene name already, type your whole gene name right here. And make sure you change this to protein, and then it will search for something for you. Or, sorry. Go on Google or something and ask Google, what does your gene express for? And that will be your protein. And then you input that protein up there. Like, hey Google, what does my gene express for? It will tell you a protein. Okay? So that's how you start this step. Alright guys, I believe most of you guys have a gene bank code or NP sequence. That works. So the next thing I want you guys to do is the following. We're going to skip the break real quick. All right. So you see how long that process took? Oh, for some of them. Okay. So now what I want you guys to do is to go to protein blast. So go in Google or something and just type down protein blast, or if you already know how to do it, then go to blast. And then in the sequence area, go ahead and type down or paste in your gene bank or NP code. Okay? And after that, you're going to search for one taxid at a time. Do not go crazy. And this is where having 4 or 5 of you guys doing it together will help speed up the process. Otherwise, if one person's doing all the work, it's going to take forever. So this is where, and also that's why you also have time tomorrow to complete this as well. So what you guys are going to do is search one text at a time, and just follow page 10 to 12. And you're going to input that taxid like right here. Like for example, the first taxid is primates. Just put down under organism right here. Put down taxid 9443, and it's just select the first one. All right, and afterward you can hit the word blast. All right, so let's do that first. So again, input your gene code, and then put in one of the taxid name, and then hit blast. And I'll wait for you guys.
Okay, how about
You guys got up here?
Yeah, we're typing it in.
All right. We're almost done. It's like the last step. All right, guys. I'm assuming every one of you guys already put this and already hit blast, right? And looking for primate. So the next thing I want to talk about now is the next concept I want to teach you guys so that you understand how to analyze your data is the following. You're gonna get a summary when you search up your code versus what you're comparing to, which is taxid code. So you're searching for Homo sapien gene, comparing it with other species. And it's going to shoot out a couple things. One thing they'll shoot out is something called a graphic summary. It looks like this. A graphic summary is just basically showing the alignment. Each one of this guy are not Homo sapien, but rather each one of this guy are all primates. Different primate species. The red, if you see call it red, that means it's very conserved. That means that gene sequence is very much aligned, very similar to one another. And then the more closer to it, to black, that means it's least conserved. That means it's completely random. It probably doesn't align. It's probably a completely different gene. It's probably analogous to it, right? All right. So the more conserved they are, like red or purple, then they must be related through some sort of common ancestry, or that gene at least came from a common ancestry, right? So you could then draw out a tree branch through evolutionary time from there. Okay. So that's how you analyze the graphic summary. So you can either analyze your data only based on graphic summary, which is perfectly fine, or you can analyze it based on the other one, which is expected value and a percent identity. That's another way you can analyze your data. Expected value is the random chance for alignment. So the lower means homolog. So the higher number, the more random chance it is. The lower the number, that means there's no randomness. It's actually there and factual. It actually aligns perfectly. So that's expected value. So the lower it is, the better alignment. The more higher it is, it's probably analogous. That means they're not related at all. Percent identity is another way you can look at. So it's right here. E value is expected value, and then percent identity is just the identity. This is the percent of the amino acid that align with each other. When it comes together, does the amino acid align to 100% or not? Right? So the higher percent, the more likely it's related. The lower percent, the more likely it's not related. And you could draw out based on the evolutionary trend. Now, you could eventually, when you do all the taxid, you eventually see a trend to where the percent identity is become lower and lower and lower, right? You eventually see that kind of trend. If you don't see that trend, there could be something very unique there, right? And sometimes, in some species, eventually when you lead to like, let's just say you're doing bacteria or plants and you're trying to link them together, sometimes it said undefined or cannot find. That's because in plants or bacteria, they probably don't have that gene, right? So that means throughout evolutionary time, it must have branched out during the animal kingdom, right? So you can tell a different story based on what you observe from there. So again, the main story here that we're trying to figure out is your gene a homolog or an analog? And what's the difference? Well, homolog is when you're similar due to inheritance. It could be inheritance based on common ancestry, based on cousins and so forth. And analog is, due to other factors. And that would be analog. Make sense? So another example of analog will be a bat's wing, how they fly, versus a bird wing, how they fly. They evolve through different ancestry and different methods. Those are analogs. All right. So your goal is to use NCBI, what you blast, one tax at a time, okay? And you're going to fill out the following information. Bar color, identity percentage, and evaluation. What you're going to do is just choose, if you look at primate, they're going to list out thousands of primates. Just choose one. It doesn't matter which one you choose, just choose one at random and fill that one out. If you look at the bar color, just average out what you see. If you see a bunch of greens, purple, and red, then the average is probably purple. Let's just say it's purple. Okay? So that's what you're going to do for all of these. You guys good?
Yeah.
And that's all I have for today.
What'd you click on?
And you're done. Hopefully you guys now know how to do bioinformatics. So congratulations, you guys are now all experts.
I don't
Good
know
job.
about experts.
Good job.
Okay.
All right. So this upcoming presentation on Thursday, it's going to be 50 points. All right? And the objective here is for you guys to utilize bioinformatic information from NCBI. You're utilizing all the data you gather, and you're creating a story from it. So your goal is to develop a hypothetical timeline to determine when a specific disease, what you've chosen, emerged throughout evolutionary timeline. When did it emerge? Did it emerge when there's plants? Did it emerge during animal kingdom? Or did it emerge since the dawn of time when bacteria still has it? Right? Each one of your genes will have something very unique. In addition to that, I want you guys to explore the potential function, advantages, and disadvantages of that particular mutation or gene in other species apart from humans. So besides humans, because you guys already have the background for it, look for the function of it on the other organism. Like, why does bacteria have a gene for Alzheimer's disease? That doesn't make sense, right? Now you guys could just, a lot of this are hypothetical, okay? A lot of it is hypothetical. So focus on the hypothetical stuff about the gene rather than providing a comprehensive lecture of the disease itself. So a lot of it is hypothetical, okay? So you might search up, like, what does this gene play for in this particular species? Even though we all have the same gene, but that same gene may function differently in other species. For example, there's a gene for jawbones in certain animals, like lizards, okay? But that gene encodes for our ear bone. Crazy, right? So that being said, the same gene here may play as a disease for us humans, but it plays as something else for the other species. And I want you guys to do research, and sometimes it doesn't show up, so just come up with some sort of hypothesis. That's perfectly fine too. I like to hear creativity, right? So you're doing that kind of research here, and seeing the evolutionary connection between all of them. Cool? So, like again, if the gene exists, it has to exist for a reason, right? Why? Like for example, why is there type 2 diabetes? Why does that exist? That's because back in the days, in the caveman years, especially the Ice Age, people who have type 2 diabetes are more advantageous than the people who do not have diabetes. Why? Because when you have a higher level of sugar in your blood, you do not freeze. If you have a low sugar in your blood, you'll freeze due to frostbites. That's why diabetes exists in the gene. So that being said, why does other disease exist? And your goal is to explain that. Make sense? All right. Good luck, guys.